thank you for the opportunity to speak. I really appreciate Tiffany giving us the opportunity to talk. I had, I think, the longest title in the program. And Brian, thanks for setting up the deal. But really what I've been working on for the last 25 years is trying to develop an ERT that can be targeted to the lysosome. Okay, and Dr. Roger explained that M6P is an important thing. So we're clever as a company. We're called M6P because that's what we focus on. So I wanted to go over a little bit about the cell physiology, about why we think this is so important. When a protein's made in the ER and it needs to go to the lysosome, you know, there's a signal on there that's called an oligosaccharide that gets phosphorylated. And that phosphorylated oligosaccharide on a lysosomal protein can then interact with the CI-MPR receptor, which Brian was referring to just as receptor in his talk. But we think this is really important because this is how a lysosomal protein naturally gets to the lysosome. Okay? This receptor is responsible for targeting most of all the lysosomal enzymes too here. But what's neat about this receptor is because it's membrane bound, it will spend some resonance time on the surface of the cell. Okay, and so I want to focus up here. So for therapies, whether it's enzyme replacement therapy or gene therapy that we're relying on cross-correction from other cells that have been transduced with the gene therapy, it needs to be phosphorylated in order to use the CI-MPR receptor, and that means cation-independent mannose-6-phosphate receptor, or mannose receptor. But if it binds here, it's also able to get internalized and delivered to the lysosome. And that's what Myozyme uses. That's the technology that Nexviazyme uses. That's what I can't pronounce that word, Brian. ATB200 uses. Kind of utilizes this receptor to get to the lysosome. Okay, but the challenge with that is this is Myozyme, Lumazyme, Alglucosidase alpha, and this is, you know, this was made some time ago. You know, we didn't know the importance and the difficulty of trying to phosphorylate these proteins in a CHO cell line. And so if you look at this, we actually have a technique that we can immobilize the receptor on a column, and we can apply any protein and see how much of it can actually interact with the receptor. If it just flows right through, it lacks MAN6P, and it can't get internalized to the cell. If it's able to bind, you know, we know that it then can act with the CI-MPR receptor and get taken up and delivered to the lysosome. One of the challenges with this early, which, you know, I'm not going to, you know, this has been an amazing drug so far, but we knew that there could be room for improvement since only about 1 in 4 molecules that we're administering actually has the proper targeting motif to get it to the lysosome. And this is really what, you know, the second generation that I heard a lot of conversation about yesterday is utilizing. They've improved the amount of phosphorylation on these enzymes to help target it. Okay. So how do these enzymes get phosphorylated? Well, there's this enzyme called glucnak-1 phosphotransferase, and it has to do 2 things. 1, it has to recognize the lysosomal enzyme, and 2, it has to then transfer a glucnak phosphate to this oligosaccharide I talked about earlier, and that's what's the targeting motif, okay? And then after it gets transferred, it has to remove this glucnak to expose that phosphate, and that's what can interact with the receptor. Dr. Roger and I have had lots of conversation about how important this covering is and stuff. It's pretty neat. But what we want to do is we really want to make sure that everything we make has this has this structure. And I showed just a monophosphorylated, but what we really want is 2 phosphates on the same oligosaccharide. Okay. But the problem with this enzyme is this is designed, you know, I put up here inefficient. It's actually a pretty cool enzyme because it's able to do this function on a lot of different proteins, which is kind of neat for an enzyme. It has a lot of different proteins it can donate to. So while it may be efficient for a single protein, it's actually pretty cool because it can do lots of different proteins to get it targeted to the lysosome. But when you overexpress one protein, it overwhelms the system and it makes it very hard to phosphorylate. And that's been the challenge with gene therapies or enzyme replacement therapies, is because you need to make a lot of this, and it kind of overwhelms this enzyme. Okay, so but fortunately, Stuart Kornfeld at WashU with Dr. Lin Liu was really trying to understand the structure of this enzyme. And I'm not going to go into details, I could. It's really fascinating. But what they did is they truncated out a couple domains to see what the effect of those domains were. And they made a smaller molecule that didn't have to have a whole bunch of processing that occurs. Inadvertently, or advertently, they made something that was 20-fold better in phosphorylating these glycans. And it works very well, okay? So that's what the technology that M6P Therapeutics is based on, is the ability of this phosphotransferase to transfer M6P to these lysosomal enzymes. Okay, so what we do is we take the lysosome enzyme of choice, in this case acid alpha-glucosidase, and we put it in an expression system that expresses the protein of interest plus this S1S3 phosphotransferase. So that means any cell that is expressing this enzyme is also expressing the S1 phosphotransferase. And what's good about this is also a membrane-bound protein, so this does not get secreted. It stays in the cell. So whether it's production in a CHO cell line or a gene therapy using virus or a non-viral, this stays in the cell. This is what gets secreted and hopefully gets taken up by surrounding cells or purified in terms of an ERT. So this is great theory, it sounds great, but what we wanted to do is take the great science and see if it actually made a difference. And I'll tell you, one of the scariest things I had to do this week or last week was show my slides to Nina before giving them. And so she told me this was too complicated. So Nina, forgive me, I did leave it in here. So because I'm really proud of this blue peak right here. Okay, we look at all the glycans. These are the glycans we release, and I won't go into details. I could talk about this one for hours. I have before, much to the chagrin of my team. But this is what we want. We want a big bis-phosphorylated oligosaccharide peak. And if I actually look at the amount of that, that constitutes over 60% of all the glycans on GAA are bis-phosphorylated. Where here is the first generation Lumazyme, and there's a little bit there, and that's probably why that's still working. Okay? But this is just the decoration on the donut. Okay? If we actually look at the donut, it actually binds to the receptor really well. Because remember I said earlier, about 1 in 4 is able to interact with the receptor and be retained by this column. Where when we take this one with the 65% of all the glycans, it binds really well. All of the molecules seem to be phosphorylated and binding well. Okay, but that's still, who cares about binding to a receptor on a column? This is how well we look at the binding. And really, when I say binding kinetics, this is how well we just measure how well it binds the receptor. And what we want is we want to be able to bind all the receptors and saturate binding, which is what this top point is. And we can see up to 100 nanomolar, we're still not saturating all the receptors. But for this one that has this huge amount of bis-phosphorylated glycans, we're able to saturate that receptor, and it works out to an affinity of about 2 nanomolar, okay, which is really nice because so little amount of the enzyme actually gets to the interstitial space. We really want this high affinity, so once it gets there, it can bind the receptor and get taken up. Okay. And it's like golf. The lower the number for affinity, the better. Okay? So that's great and that's wonderful, but we really want to see how it works in vivo. How does it actually work in an animal? So, sorry. I can go back. All right. So what we did is we did 20 mg per kg every other week for 6 months. And I'm just showing you one of the things we looked at, and we looked at grip strength. Okay, this was early on. We were learning how to do it, but what we were really excited about was that we start off with Pompe animals that have lower grip strength, and we compare this to wild type. And we think this is some of the measurements that we were learning. But as you see throughout the life of a Pompe animal, or the 6 months of a Pompe animal and a wild type animal, the grip strength is lower for the Pompe animal throughout that. On Lumazyme, you get some improvement, but it's not as much as we would like. Okay, what we were really excited about is by like 3 months, we were able to normalize that grip strength in the Pompe animals. And so we were really excited about that, but is that real? So we decided to go take a look at the tissue. Okay, so we looked at glycogen levels in the heart, quad, triceps, and diaphragms. And what I would focus in on is the skeletal muscle, which seems to be more, what we want to focus on in the adults. And we actually were able to seem to be really close to normalizing glycogen in this 6 months of therapy or 12 treatments. And we were really excited about it because that kind of matched up with the grip strength data that, the biochemical reduction in glycogen did seem to have a nice phenotypic. Now, this is the slide I may get in trouble with Nina. So we were really encouraged by this, and so I'm going to build up to the next slide. So we've done this. This is early. This is preclinical. I'm not proposing this is the final how this works. But during COVID, we had trouble shipping some of our drug to some of our CROs that were running the animal studies, and they skipped doses. But it didn't seem to change the grip strength data. So what we decided to try, and that yesterday after hearing about the dosing at 40 mg per kg every week, that's actually kind of done in clinic, we decided what happens if we do a debulking strategy, get really aggressive at the beginning of therapy, try and reduce glycogen as much as possible, and then go to a once a month dosing. And we compare that to a twice a month dosing or every other week. And so we did the grip strength again, and what we saw is that by that 3-month period of therapy in a 6-month-old mouse, that we were able to get the same type of grip strength reduction following this debulking, and then we were able to maintain that with just once a month dosing, which we thought was kind of neat. So maybe we are getting a nice phenotypic correction of some of the cellular pathology that isn't right on the edge, so it can withstand GAA not being there at the full level all the time. So, and I'd like to say that this work that I'm showing now, I'm very thankful to the AMDA Foundation because they're helping fund this work. They're helping us do this. Oh, okay. So really good because I have one slide. So I have a summary. Okay, so basically what we're doing by putting this type of construct where we have the enzyme plus the S1S3, we're able to get better CI-MPR binding. That seems to help with the more efficient glycogen reduction, and it seems to normalize the grip strength. And so we think this is something that can apply to ERT, gene therapy and stuff because we are co-expressing them. And with that, I'd like to say thank you.